Kristen, and she is allowing us to record her. Let's continue that. So Kristen has been at the University of South Carolina in Columbia for a little while. She received her master's degree working with Dr. Charles Maticus, also received her Ph.D. With him, and then moved on to a postdoctoral fellowship working with Rosemarie Booz. She's got a number of fellowships that have been awarded to her throughout her academic training career thus far, and has secured a couple of small grants during her postdoctoral work, so kudos. And I think her publication record is very impressive. She lists 25 publications to date, with others that have been submitted and others that are in the works. And for a postdoctoral fellow with her years of experience, that is an extremely high level of productivity. It's very interesting work published in good journals. So with that, I will pass it over to Kristen.
Thank you for that wonderful introduction and for providing the opportunity for me to speak about some of my dissertation work. So the work I'm going to speak about today is entitled Progressive Neurocognitive Impairments and Neuronal Injury Resulting from Chronic HIV-1 Viral Protein Exposure. So as we heard in the previous talk, HIV-1 associated neurocognitive disorders, or HAND, persist despite treatment with combination antiretroviral therapy. Specifically, HAND afflicts between 40 to 70% of HIV-1 seropositive individuals. Cross-sectional studies have been instrumental in defining the primary neurocognitive domains that are altered in HAND, including deficits in information processing speed, memory, attention, and executive function. The work I'm going to share with you today is focused on the primary neurocognitive domain of attention. More recently, longitudinal studies have revealed that a significant proportion of individuals exhibit overall decline across time. However, to date, there is no extant in vivo biological system that's able to model the longitudinal progression of HAND in the post-CART era. The pathogenesis of HAND in the post-CART era is multidimensional and may include neurotransmitter alterations, synaptic dysfunction, and neuroinflammation. However, functional alterations in neurons more likely underlie the pathophysiology of HAND and have significantly been correlated with cognitive deficits. These functional alterations may be characterized by dendritic injury, synaptodendritic dysfunction, and alterations in dendritic spine connectivity and morphology. However, there remains a critical need to determine whether these functional alterations in neurons progress across time. Thus, today I will be presenting two key studies examining progressive deficits. The first study examines the progression of neurocognitive impairments across the functional lifespan. The hypothesis is that there would be a differential progression of neurocognitive impairments in HIV-1 transgenic versus control animals. Our second study will utilize a time sequential longitudinal experimental design to examine neuronal injury and synaptodendritic alterations in pyramidal neurons from layers 2 to 3 of the medial prefrontal cortex. It was hypothesized that these alterations would progress in the HIV-1 transgenic rat across time. As I've alluded to, these studies will be conducted using the HIV-1 transgenic rat, which was originally reported in a seminal paper in 2001 by Reed et al. The HIV-1 transgenic rat contains a GAG pol-deleted HIV-1 provirus that's regulated by the human viral promoter. This renders the HIV-1 transgenic rat non-infectious. However, viral proteins are expressed constitutively throughout development, resembling HIV-1 seropositive individuals on CART. To date, there is growing acceptance and confidence that the HIV-1 transgenic rat is a valuable tool to model key aspects of HAND, as evidenced by numerous publications from our labs and others. The first study that I will share with you today focused on establishing the progression of neurocognitive impairments across the functional lifespan. In order to be able to establish the progression of neurocognitive impairments, we must first establish the integrity of sensory and motor system function. This was examined by conducting pre-pulse inhibition of the auditory startle response, as well as locomotor activity repeatedly throughout the functional lifespan from postnatal day 90 through postnatal day 480. We first examined body weight as an index of somatic growth. We can see that HIV-1 transgenic animals, independent of biological sex, weighed significantly less than control animals. But both HIV-1 transgenic and control animals exhibited steady growth throughout experimentation. With regards to the integrity of sensory and motor system function, today I'm going to focus on the data collected at postnatal day 480. Specifically, in the panel on the left and the center panel, I want to focus your attention on this robust inhibition that is observed following the presentation of a punctate auditory or visual pre-stimulus. The robust inhibition supports intact sensory system function. In addition, we conducted locomotor activity as a way to examine gross motoric system function. We can see that both HIV-1 transgenic and control animals exhibited a significant number of photocell interruptions. In fact, at postnatal day 480, HIV-1 transgenic rats exhibited a significantly greater number of mean photocell interruptions relative to control animals. Thus, there is no evidence for any motoric impairments in the HIV-1 transgenic rat. Now that we've established the integrity of sensory and motor system function, we can examine the progression of neurocognitive impairments using repeated assessments across the functional lifespan. Today, I will focus on our assessment of sustained attention and stimulus reinforcement learning. However, we also examined pre-attentive processes, long-term episodic memory, and measures of executive function, including flexibility and inhibition. During phase 1, which was original acquisition, our animals were trained to lever press using a shaping protocol. They were then trained on a signal detection task, which I will explain in more detail in the following slides. Our final component was to vary the duration of the signal from 1,000 to 500 and 100 milliseconds. This afforded an opportunity to look at the temporal components of sustained attention. Original acquisition was conducted at approximately 2 months of age. To complete the task, animals were required to meet 70% accuracy for 5 consecutive or 7 non-consecutive days. The animals were then retested every 60 days after the successful completion of original acquisition through 6 retest assessments. We can see on the screen a schematic of our operant chambers. So we place our animals in the chamber, and on the front wall, there's 2 levers, which are indicated in green, a magazine where the sucrose pellet reward is presented, and there's 3 panel lights. We specifically want to focus in on the center panel light, which will be utilized as our signal during signal trials. So the animal is focused on this front wall of the operant chamber, much like a human would be examining the computer screen. So we're now going to focus in on this front wall and how an animal will be seeing the experiment. So during a signal trial, the center panel light was illuminated and the levers are presented. If an animal hits the right lever, it's considered a hit and they are rewarded with a sucrose pellet. However, if an animal hits the left lever, it's considered a miss and the animal is not rewarded. We also have non-signal trials whereby the center panel light is not illuminated. The levers are, however, presented, and if an animal hits the right lever, it's considered a false alarm and the animal is not rewarded with a sucrose pellet. However, if the animal now hits the left lever, it's considered a correct rejection and it's rewarded with a sucrose pellet. Thus, the animals must attend to this center panel illumination throughout a series of 162 trials during the session. During the original acquisition of signal detection, presence of the HIV-1 transgene failed to significantly alter the number of days to criteria, which is illustrated in panel A. The number of days to criteria is an index of stimulus reinforcement learning. However, I specifically want to focus in your attention on panel B. In panel B, what we see is the number of hits and the number of misses that the group of animals has across signal durations. So this is the data that's presented from when we're varying our signal durations. We're specifically interested in this point of intersection, which indicates where our animals lose the ability to accurately detect the signal. We can see that again during original acquisition, presence of the HIV-1 transgene failed to significantly alter the temporal components of sustained attention. The yellow line indicates the loss of signal detection for the transgenic animals, and the green line represents the loss of signal detection in control animals. When we look at retest assessments, we can focus in on these two lines. However, first, I want to examine the progression of stimulus reinforcement learning. Control animals displayed significant continuous improvement in task acquisition across retest assessments, reaching asymptotic performance at approximately 7 days. However, in sharp contrast, HIV-1 transgenic animals exhibited an initial improvement in task acquisition, followed by a progressive decline. Thus, these alterations suggest a progressive deficit in stimulus reinforcement learning in the HIV-1 transgenic rat. We will now examine the progression of the temporal components of sustained attention. First, we have retest assessment 1, where our HIV-1 transgenic animals are impaired relative to our control animals. What we see is that the repeated assessment of sustained attention across time allows both HIV-1 transgenic and control animals to improve their performance. However, our HIV-1 transgenic animals improve to a lesser degree and fail to improve as quickly as our control animals, thus supporting a progressive deficit in the temporal components of sustained attention. Thus, our conclusions from study 1 are that even in the absence of sensory or motor system deficits and comorbidities, hand is characterized by age-related disease progression in temporal processing and long-term episodic memory, data which were not shown in the present talk, as well as learning and sustained attention. However, there remains a need to examine the progression of potential pathophysiological mechanisms underlying hand. Thus, in the second study, we examined the progression of neuronal injury and synaptodendritic alterations using a time sequential longitudinal experimental design from postnatal day 30 to postnatal day 180. We were specifically interested in examining pyramidal neurons in the medial prefrontal cortex. Animals were sacrificed every 30 days, and a ballistic labeling technique was utilized to be able to see our pyramidal neurons. They were analyzed using Neurolucida 360, which is a sophisticated neuronal reconstruction software that allows us to examine aspects of neuronal morphology and dendritic spine morphology. With regards to neuronal morphology, I'm going to focus today on the measurement of dendrite length. In control animals, we see an initial exuberant number of branches and therefore dendrite length that are pruned during adolescence, reaching stable levels in adulthood. However, our HIV-1 transgenic animals exhibited a differential developmental trajectory evidenced by a linear increase in dendrite length across time. The schematic on the right illustrates what we see in our neurons, whereby at postnatal day 30, our control animals exhibit exuberant branching that's pruned and reaches stable levels in adulthood. However, our transgenic animals, which are shown in panel B, exhibit abnormal neuronal development that's characterized by this excessive branching at older ages. We then examined the total number of dendritic spines as a measure of synaptogenesis. Specifically, dendritic spines may be an index of excitatory synapses. Again, and consistent with previous literature, control animals exhibit an initial excess in the number of dendritic spines that are pruned during adolescence, reaching stable levels in adulthood. However, HIV-1 transgenic animals exhibited a prominent alteration in synaptic pruning, evidenced by a linear increase in the total number of dendritic spines across time. And finally, we examined dendritic spine morphology. I'm going to present the results from dendritic spine volume, which is correlated with the area of the postsynaptic density and may be indicative of synaptic function. Specifically, we see that in control animals, dendritic spine volume increases in a linear manner from postnatal day 30 to postnatal day 180. However, in HIV-1 transgenic animals, dendritic spine volume decreases in a linear manner from postnatal day 30 to postnatal day 180, supporting a progressive decrease in synaptic function. In addition, across time, the HIV-1 transgenic rat exhibited a progressive increase in dendritic spine backbone length and a progressive decrease in dendritic spine head diameter. So we've now added on to the schematic that I presented earlier, whereby again, our control animals exhibit an exuberant number of dendritic spines at postnatal day 30 that are pruned and become these mature spines that could be potentially classified as a mushroom phenotype. However, in the HIV-1 transgenic rat, these more mature spines regress across time and exhibit a more immature phenotype at postnatal day 180. It is possible that the exuberant number of dendritic spines and dendritic branches observed at postnatal day 180 are a compensatory mechanism for the dendritic spines being more immature relative to control animals. Thus, our overall conclusions are that even in the absence of sensory or motor system deficits and comorbidities, hand is characterized by age-related disease progression. And second, that neuronal injury and synaptodendritic alterations, which are key neural mechanisms underlying hand, are observed early in the course of HIV-1 viral protein exposure and progress through 6 months of age. The data I've presented today were parts of my dissertation, and my goals as a postdoctoral fellow are to expand my expertise to include substance use disorders with an emphasis on opioid use disorder, given its comorbidity with HIV, as well as to master novel neuroanatomical methods, including in vivo fiber photometry, AAV gene delivery, and stereotaxic surgery for the evaluation of neural mechanisms, and to apply advanced data analytic approaches to establish causal neural mechanisms. With that, I would like to acknowledge Dr. McTutis, my graduate mentor, Dr. Booz, my postdoctoral mentor, our collaborators, our research associate, and our funding sources. Thank you.
Great talk, Kristen. Thank you very much. One question in the chat so far. I have some, but we'll begin with Diane. Oh, that's to me. Oh.
Norm, that was to you.
Okay, that's a question to me.
Sorry.
I'm sorry. So actually, Kristen, could you clarify one thing for me? I wasn't quite sure I understood this. So the HIV transgenic animals learn slower, they're not as complete, but yet the number of errors and the number of correct responses is not different. How does that work? Yeah.
So we train them to the same accuracy. So even during retest assessments, we're training them to 70% accuracy. And so to be able to complete the task, they have to meet this criteria. So it's just it's likely due to our stringent criteria that we established from the beginning.
Okay. I think one of the initial figures you showed, I think, was how long it took them to get to criteria, and that looked similar as well. So is this initial learning deficit sort of compensated for later on?
So I think it's a kind of an age-related progression. So what we kind of see is that, yes, initially they are very similar and they don't exhibit deficits, but then as we retest them more, they do exhibit deficits whereby it takes them significantly longer. And if we look at those curves, which we can look at for each retest assessment, they shift farther and farther apart. And so what happens is our curve becomes, the slope decreases such that it's taking some of those transgenic animals an extremely long time. We are retesting them every 60 days. So what happens is that if an animal takes 30, 40 days, it's only not being run in the task for another 30, 40 days or another 20 days to meet that 60 again. So these are some of the confounds because of the repeated assessments.
Great, thank you. That helps a great deal. It clarified things perfectly. Let's see. Heather Thomas, I'm not quite sure who this is, but please unmute yourself and go ahead and ask your question. Is there a deficit?
I think it's Pim.
Oh, it's me? Okay, all right, fine. So I'm from NIMH and I'm actually a pediatric neuropsychologist. And so my question is whether the deficit that you're seeing is not a deficit in the ability to benefit from practice, rather than it is a progressional deficit. This is something that we very frequently see in kids that basically have a deficit, and that if you look at their raw scores, they continue to decline or to have a larger difference from a normally developing kid that is learning more, but basically their IQ stays the same.
I think that's very similar to some of the things that Dr. McTudis and I have talked about is the question of practice effects. And so at some level, we may not be seeing the full deficit because our animals are able to repeatedly practice. In fact, now we're looking at older animals that are approximately 14 months of age that have never been assessed before to be able to see what's going on with that longer constitutive expression in the absence of previous assessments.
Okay, can I ask a question?
Of course.
Sorry. Hi, Kristen. This was awesome. I love your study too. It's really, really nice. But I want to ask one question about your behavioral assessments. How much could that be influenced by the fact that the animals are progressively blind? They are born with a cataract and they get progressively blind over time. And so this should start around 5 months of age. They also have several multi-organ disorders that could account for sickness behavior. So I want to know how much of these factors could influence your behavioral assessment. Thank you.
Thank you. So in our hands, we've examined both auditory system function, gustatory system function, and visual system function. And specifically, our focus has been on visual system function for brightness, because that is what is required to complete our attention task. And we see that even through advanced age, they are able to detect brightness. So we don't believe that that is confounding our results and our findings with regards to neurocognitive impairments. We haven't actually evaluated acuity, but that is not really required for our task. In addition, we don't see any evidence of gross motoric system function alterations through 18 months of age. So we know that that was not a confound in our study as well.
Thank you.
Thank you.
We have a question in the chat from Jacques Baker. If you wouldn't mind unmuting yourself to ask the question.
Sure, I was just, I might have missed it, but was this in males only? And if it is males only, could you speculate on what you think you would see in females?
So sorry, I didn't clarify that. This was in both males and females. We see very similar things in both males and females. So the data I presented were actually collapsed across biological sex. So yes, we see very similar progressive deficits, but that has been integral to our experimental design in both studies.
Thank you.
Thank you.
And Dr. Dash has a question. Prasanta Dash, oh, who has microphone issues. So I will ask the question. You mentioned that you looked at an effect on hand, but the study was done in just TAT transgenic animals. How can you extrapolate, interpretate these data into the real context? Because these animals can't be administered with ART to look at what happens, as none of the ART drugs are anti-TAT.
So our study was conducted in the HIV-1 transgenic rat, which expresses 7 of the 9 HIV-1 viral proteins. So it has TAT, GAG, NEF, the envelope protein, and others. It is missing GAG and POL, so it is non-infectious. We don't treat with CART, but to date, there's mixed evidence as to whether CART affects neurocognitive impairments. There's limited evidence that it affects dopamine neurotransmission.
Great, thank you. And I'll ask one last question unless somebody else has one. So it looked like there were, in some instances, and I'm thinking of the, I think it was the number of synapses, the baseline differences were quite different between the transgenic, non-transgenic animals. When do these proteins start to get expressed in the rat? Do they get expressed in utero?
So what happens is that we often breed a HIV male and a control female so that we make sure that the female mother is mothering the animals properly. Some of the rats have cataracts, and those are the ones we use because we know that the transgene is in the correct place. We've seen neurocognitive impairments as early as day 14. So we definitely believe that they're having very quick effects. As demonstrated, yes, we saw alterations at postnatal day 30 in the brain.
Thank you.
Thank you.
And if there's any other questions, just please unmute yourself and go ahead and ask.
So I have one more question. So one of the things that is obvious in the adult human population, as well as in the kids, is the large heterogeneity. So how consistent were the data across all your animals?
I think that was one of the really interesting things about our examination of these animals, especially with the progression of learning. I'm going to try to share my screen one more time. Because I think this is something that I find very interesting and is fascinating to me personally. Because what you see is that across time, we do see an increased error. And so we do see heterogeneity within our rats. We've also seen this when we treat them with methylphenidate, such that some of the animals will respond, but some will not. So it's definitely something I'd be really interested to look at, is the individual differences in these rats and whether there's ways that we can predict which ones may have greater neurocognitive impairments.
Thank you. That's really, really interesting. Thanks for sharing that.
Thank you.
It is, thank you. And thank you everyone for the questions. So we'll just close out by thanking our 2 speakers today for 2 outstanding talks. Very much enjoy hearing trainees speak. It's the most contemporary work that's out there. And next week we are on, same time, same place. We have Dr. Gary Schroesdeck, who is a world-renowned metabolomics expert, has developed a lot of the metabolomics methods that are out there, who will be talking about the use of those methods in neurodegenerative diseases with an emphasis on HIV, of course. So stay safe, everyone. We'll see you next week.